Hi, my name is Artem Babaian. Today I'm going to be sharing with you my work on Petabase-scale sequence alignment, catalyzing the discovery of novel coronaviruses. It doesn't take a lot of motivation to convince you that it's important to study coronaviruses right now. The entire global research community has been analyzing now over 1 million SARS-CoV-2 genomes. And so while the global community is focusing on what's unfolding in front of us, me and a group of collaborators decided, how about we look exactly the other way? Can we understand where this virus came from in evolutionary terms? And this is actually an important problem. SARS-CoV-2 is biochemically predicted to infect over 80 species of mammals. So if this virus enters one of these mammals, it can recombine with some of the native coronaviruses. Now, when this recombined virus, say, changes its spike protein and re-enters humans, this actually leads to a very real risk where this virus will be able to reinfect humans, and the herd immunity that we've worked so hard on can be lost, which would pose quite a danger to us. You know, this was just a prediction early on, but now we're seeing that this is actually happening. So there were tigers that were infected with SARS-CoV-2 in Knoxville and Brooklyn, dogs and cats in Ontario and China, and then a very tragic case of minks in Denmark where the virus infected minks and then went back into humans, leading to the culling of millions of animals. And so we decided, okay, what if we were to just analyze everything, every sequencing data set that contains RNA? And very fortuitously, back in February of 2020, NCBI mirrored the sequence read archive onto AWS S3 systems. This gives us an unprecedented level of access to massive amounts of data. How big? Well, one petabase is 1 followed by 15 zeros. And currently, there's over 50 petabases, which is roughly equivalent to about 100 petabytes of sequencing data. And this data is growing exponentially. And so while the availability of data is just exploding, outpacing Moore's law even, our ability to access that data in a meaningful way is falling behind very quickly. And this is where cloud computing can actually really excel. And this gives us access to massive amounts of data. We're talking every corner of the planet, from cell lines in Vancouver to anal swabs of penguins in Antarctica. All of this data is right here at our fingertips if we can analyze it. And so, we did this as an open collaboration as part of this hackathon, HackSeq-RNA, that happened in Vancouver in May of 2020. And so what we came up with was a Ceratus architecture. And the main idea here is to go really, really big, you have to first go small. And so we aggressively cost optimize this architecture to process petabytes of data and gives us access to over 22,000 virtual CPUs at once. So there's a few kind of key points that I'd like to share with you that will apply to pretty much anyone doing bioinformatics research. So the first thing to consider is that alignment is what is called an embarrassingly parallel problem, meaning that every read pair can be aligned or processed on the CPU independently of every other read pair. And so what we do actually is, instead of trying to work with very big files that can reach, say, 100 million reads per file, we break that up into very uniform blocks. We're calling FASTQ blocks. And we do the work on these FASTQ blocks. Now, the way that I'd like you to think about this is that this creates an atomic unit of work. So when you have a variable file size, you have to essentially engineer your pipeline for the worst case scenario. Otherwise, the big files will fail via errors where, there's not enough disk, there's memory issues, or the runtimes are too long. And then if you engineer your pipeline for that, for the kind of the worst case scenario, then you have a lot of slack when you have these smaller files. Whereas if you have a uniform workload, it's very predictable and reliable, the CPU usage, the memory usage that you expect from every atomic block. And this allows you to create a reliable cluster, which overall means that your efficiency will go up significantly. So overall, what does this look like? Well, for the alignment modules, we can access just over 4,000 instances, and we either use Bowtie 2 for nucleotide alignment or Diamond for translated nucleotide alignment. Everything runs on individual threads, which means that we have over 16,000 alignments processing all at once. This is moving 100 gigabytes of data a second. So what does this mean at the end of the day? Well, using AWS, Ceratus can process in excess of 1 million sequencing libraries per day. The cost per library is less than 1 cent. We are talking about massive, truly petabase scale sequence alignment here. And here's just a breakdown of what that looks like. In 24 hours, we analyzed 1.29 million libraries. And if you look at the breakdown of costs, you can see that the majority of the cost is focused on that computing, which is where you would want your costs to be. By having these small units of work, we're able to essentially eliminate conventional block storage or movement of data that would be associated with that. So what did we actually do biologically with this? So very quickly, we realized that we don't have to just focus on coronavirus. We actually studied all RNA viruses. So from public sequencing databases, we identified 15,000 species of RNA viruses defined by 90% amino acid identity of the hallmark gene RNA-dependent RNA polymerase. You can think of this as kind of the heart of an RNA virus. And so we classify these based on their catalytic cores. And using these sequences, we're able to analyze 5.7 million sequencing data sets, or over 10 petabases of sequencing data. You can see that the global distribution of RDRPs covers every corner of the world. The oceans, the mountains, deserts, where they're sequencing data, there are RNA viruses. So what's the punchline here? In 11 days, we found over 130,000 novel species of RNA viruses. To put that into perspective, that increases the total known RNA viruses found over the last 130 years of research by an order of magnitude, and that was done in under 2 weeks. So bringing it back to coronaviruses, well, you do an unbiased analysis to find things that you don't expect. So we were able to identify 9 new species of coronavirus, and the most interesting ones were associated with aquatic vertebrates. So here we have axolotl, seahorse, pipefish, fugu fish. There was a similar virus identified in Pacific salmon, and these viruses cluster. And one thing that was a kind of a paradox and bugged us for the longest time is that we were never able to identify the spike protein on these coronavirus genomes. We would always get the RDRP segments. What actually happened was that these viruses appear to be segmented. So they occur on 2 separate molecules. You know, if you think of SARS-CoV-2, the genome is one RNA molecule. But these groups of coronaviruses, they occur likely on 2 different molecules. If you think of where segmentation also occurs, influenza is another place where this will occur. With influenza, these segments can reassort or kind of swap out between different viruses. Now we understand that this could actually also happen in coronaviruses. This doesn't pose any kind of threat to humans because these viruses are quite diverged, but it does tell us something very important about the basic biology of coronaviruses. We make this assumption that it always have to be on this one molecule, but that's not true in evolutionary time. And the other way that we were able to analyze this data is to look for discordant libraries, where you have the coronavirus and then you're finding it in a place you wouldn't expect to find that coronavirus. So we found a bunch of coronaviruses in plant samples, probably because, you know, say like a pig pooped and that was fertilizer for the corn, and so the virus was found on the corn sample. We had a very interesting case that you don't expect to find these. There were 2 children sequenced in 2010 who had fever of unknown origin. Within their blood, there were nucleic acids for murine hepatitis virus, which is a beta coronavirus that infects mice. So normally you would not expect a mouse coronavirus to be found in humans, but here we have 2 children where this is the case. This actually leads to a very interesting possibility where this type of unbiased metagenomic sequencing can be a very important diagnostic for monitoring for spillover of viruses into humans. And so instead of waiting for this to amplify and affect many people, you can identify these spillover events very early on and then act on this information quickly, where you can contain it from a public health perspective before you allow for that exponential growth to kick off. One caveat here is that the presence of nucleic acids do not prove that infection has occurred, so it only is implied. So we don't know if this virus actually infected these individuals. In addition to coronaviruses, we focused on the poorly characterized Deltaviruses. These tiny RNA viruses contain 2 ribozymes and 1 protein antigen, and their circular genomes fold into characteristic rod-like shapes, shown here as base-pairing arcs. The best-known example is human Deltavirus, the causative agent of hepatitis D. There were 10 known Deltaviruses. Using Ceratus, we identified over 350 related viruses. Some of these had similar genomes to the known Deltaviruses, including the Marmota monax or woodchuck Deltavirus. Woodchucks are widely used as a model organism for hepatitis B-induced liver cancer, which may now be confounded by the presence of an additional virus. Perhaps most excitingly, though, there were distant viruses we're terming Zetaviruses. These elements have tiny genomes, ranging from about 300 to 800 nucleotides long. Zetaviruses contain 2 complementary ribozymes and an overall characteristic rod-like folding akin to Deltaviruses. Unlike Deltaviruses, they do not encode a single protein. Instead, they have 2 open reading frames covering the entire genome on both the positive and negative strand, often lacking stop codons. This leads to potentially endless in-frame translation of these proteins, all while maintaining genome complementarity for rod-like folding. So while we know little about the biology of these elements, they show a hypercompression of genetic information that can only be described as elegant. If you'd like to learn more about these new viruses and their ribozymes, please check out Talk 170 by Amelia Cervera. I'd like you to come away from this with one main idea. That is, we are entering a new era of petabase-scale sequence analysis. Over the past decade, we all have deposited our data into the sequence read archive, brick by brick, and it is now the moment to step back and view this biological cathedral in its entirety. At our fingertips are literally millions of biological samples making up a digital history of Earth's genetics, and this sequencing representation of our planet is lighting up exponentially. The order of magnitude increase we show here with RNA viruses is at best half of what will be available next year, and it is only a tiny sliver of the overall genetic diversity available to all of us. All of the Serratus data is released into the public domain immediately. I strongly believe the SRA and our derived data are a common resource to be shared with everyone. If you have any interest in exploring novel viruses, please visit serratus.io or reach out and I'd be happy to help. I'd like to truly, deeply thank all the contributors that have helped make this project possible. I'd like to also give a heartfelt thanks to the UBC AWS Cloud Innovation Center and everyone there. Thank you all for your time, and I hope that soon we could see each other face to face. Take care.